away from most of the topics, but as today we covered already air pollution, soil pollution, I will talk about water pollution. And so this is a part of our big experiment, a study on the effect of water pollutants, of xenobiotics on fish gametes, on fish reproduction. And today we'll talk about in vitro effects of bisphenol A on DNA integrity and intracellular signaling in spermatozoa of sturgeon. So we know that nowadays more and more pollutants come to the water, to the surface water. Such as bisphenol A, which come from plastic, and its concentration they vary hugely, and for example like 0.1 microgram or some nanograms per liter in non-polluted waters, up to 2 milligrams per liter in very polluted areas. And of course this, like throwing bottles in the river, it's not the main source of pollution, but factories and production and shipment and so on. And a lot of these pollutants, other pollutants also, they come to the water and affect aquatic organisms, like fish, for example. And one of the adverse effects of these pollutants is disruption of endocrine system. It's impairment of, in particular, impairment of gamete function and reproduction. What is interesting in fish is that most of fish species, especially freshwater fish species, they are externally fertilizers. So they spawn eggs and sperm into the water, and in just few seconds that they have, the cells must operate and they have to fertilize. They have to fuse and fertilize to produce embryo. On this slide you can see this our model organism. This is a small sturgeon or starlet. It's an endangered species, which populations in wild in nature are now decreasing due to overfishing and in part due to water pollution. But it's also commonly used in aquaculture. Maybe you know this is quite famous for production of meat and black caviar. Our question was how water pollution can affect spermatozoa, so male gametes of this fish. And in particular, how bisphenol A affects sperm motility, DNA integrity, and protein phosphorylation pattern. To assess this, we collected sperm from several males, incubated it with BPA in different concentrations for 1 hour. We assessed sperm quality with few different techniques. So we recorded motility of the cells and calculated the percentage of motile cells, percentage of motile spermatozoa. Previous speaker was already talking about cometa-assay and what it shows, so I will just say that we analyzed DNA fragmentation by percentage of DNA in comet tail. And we assessed a little bit intracellular signaling with Western blotting using anti-phosphoserine antibodies and antibodies against protein kinase C substrates. So here you can see that the percentage of motile spermatozoa was determined in 15 seconds after triggering motility, so very fast. And you can see that the percentage of motile cells decreased, and even at 1 micromolar concentration of BPA decreased significantly compared to control, and in 10 micromolar it decreased like up to 15% or even low compared to control. Also DNA integrity was affected by bisphenol. So in high concentrations we can see the significant difference of DNA, percentage of DNA in the common tail compared to control. Intracellular signaling was affected as well. So you can see that phosphorylation pattern on the serine residue changed into mainly on two protein bands. One of them was dephosphorylated, and approximately 100 kilodalton protein band was dephosphorylated, and another band, about 230 kilodalton, was phosphorylated in the highest concentration of bisphenol. Also, it affected, this compound affected phosphokinase PKC. So this is quite important kinase for sturgeon spermatozoa, participates in motility activation. And you can see that this band, 100 kilodalton, is dephosphorylated compared to control to incubation. So I will a little bit explain here more mainly about spermatozoa, because I see that nobody worked so much with sperm. So oh, you can see. So you can see this is a sperm cell. Head, midpiece with mitochondria and tail or flagellum, which contains axoneme, and this is motor of the cell. So here this thing it produces bands during motility, and these bands propagate the cell effect, the movement. So the PKC, as we showed in our previous study, it's important participant in this motility activation. It phosphorylates dyneins along the flagellum, and dyneins they create, they start to, let's say, bend the flagellum. They move microtubules. Microtubules start to move between each other, and the wave is created. So the effect of bisphenol in this case, it shows that it, in particular, it affects can affect intracellular signaling and affect motility through this changes of kinase activity. And this is quite well-known scheme. I think that endocrine disruptors such as bisphenol A, they can bind to nuclear receptors and induce transcriptomic response, or they can bind to membrane receptors and change the activity of kinases, phosphatases, calcium signaling, and in perspective, like in affect mitochondria function as well. In our previous studies, we showed that in the fish spermatozoa, since they are transcriptionally inactive cells, so there is no transcription and no like this we can study. But we showed that endocrine disruptors induce reactive oxygen species production. So it induces oxidative stress, which affects DNA integrity, lipid oxidation, protein oxidation. It alters mitochondria function. And this was published in 2013 by my colleague. And in this analysis, the last studies, we showed that it also can influence this pathway, so kinase and intracellular signaling pathway. So, in a very short conclusion, I would like to say that yes, bisphenol is, again, I think this is what many people say, bisphenol is dangerous. Bisphenol is can, at least in our tested concentrations, it was bad for sturgeon sperm and for sturgeon gametes. Decreased sperm motility, and of course decreased sperm motility represents like then part of the effect. It shows that these pollutants can affect also not only like individual fertility, but possess problems for all fish population. And damaged DNA can be transferred then to the offspring and induce problems in embryo development, in growth, and so on. For aquaculture, this is could be quite a problem, and for wild populations, of course, as well. On the other hand, there's another point in our study, because this shows that fish spermatozoa are very sensitive to the presence of pollutants. These cells, you saw the cells, they're very tiny, and they almost don't have cytoplasm, and they almost don't have antioxidant protection. Which means that compounds, external like nanobiotix, they're quite effective for them. They can have a big effect, and we can monitor this effect quite easily just to check the spermative. So this is like one of the possibilities to use this the cells for monitoring water pollution or to assess aquatic the effect of pollution for aquatic organisms. So here I would like to say thanks for our Research Institute of Fish Culture and Microbiology, for all my collaborators, especially for projects that supported this study and my stay here. And of course I would like to say thanks for conference organizers for this nice possibility to come here to Valencia in this nice spirit of summer. And thank you for your attention.
Thank you very much. One question? Yes.
Why you used these concentrations exactly?
We selected these concentrations because they had like a most prominent effect. If you read our other studies, we also tried to use concentrations that were, let's say, environmentally relevant. So the concentrations that occur in water, that were reported to be to occur in water, and we tried these concentrations. For them, you have to incubate longer, and the effect is less prominent. So this is just to show that it's like a model, yeah.
And what you used in context, percentage of DNA only?
Just to show that there is some DNA fragmentation. So it's just
to see that's enough?
Depends on what you want to show. Yeah, like if you want to go deep in this, and if you want, for example, I don't know, 2-strand breaks and so on, yes, you can use. But I guess percentage of DNA in tail is more than enough.
Are there literature review in this used only percentage of DNA in tail?
There are quite a lot of studies that use only this one parameter.
Okay, one question?
Yes, I have a question. When you saw your mechanism, the mechanism of action of these xenobiotics, you saw a membrane receptor. Do you know which one is?
No, we don't know for the moment. Unfortunately, we don't know for the moment, and this is like a problem because fish spermatozoa are quite specific cells, and so we need to make a study to find which receptor it can be.
Okay, but
there are
in the literature you can look for that.
Yes, but not so many literature.
Yes. But xenobiotics and the gardeners in connection with DNA expression and is the aryl hydrocarbon receptor.
Probably,
I think
but
so.
we need to find it in fish sperm first. Like there are not so many studies on fish sperm and on fish sperm proteomics. Like not so many studies reported. Yes, there is this receptor and this receptor and this receptor.
I was in Belgium. I did my master degree in Belgium, also in application with fish, you know, and testing antibiotics, and also there were some works because it's in fact a problem.
Yes, we also actually we tested few different pharmaceutical compounds like oh, I forgot the name. For example, antihistamines. They work with the sperm perfectly, like they stop motility. And
Really?
and who guessed that in fish sperm there are histamine receptors?
And did you look for information because in humans should be the same, or?
No, unfortunately.
It's
Because
different?
it's different because humans are internal fertilizers. So there is absolutely different scheme of motility activation. So that you can say, okay, it can run and it has nothing to transfer, the DNA can. So, like in this case, but
It will be interesting to test which type of medications in humans they affect the mobility of the sperm cells.
no, there are kind of